Before we get started on the review for heredity today, there's a couple of things that I'm not going to go through with you guys today, and that's going to be some of the basics of like meiosis and mitosis and meiosis. For the most part, I'm hoping you guys can study those and remember that their odds of them asking, hey, what's the difference between mitosis and meiosis is going to be pretty slim. For the simple fact that that's more memorization than it is actually application. And because it's open note, they don't want to do any memorization. So just a couple of quick things. Meiosis is a process that ensures the formation of haploid gamete cells in sexually reproducing diploid organisms. Meiosis results in daughter cells with half the number of chromosomes of the parent cell, and meiosis involves 2 rounds of a sequential series of steps, meiosis 1 and meiosis 2. Now, a FRQ question could ask about, hey, what happens if this goes wrong in meiosis? And that's a good reason to study it. It's just that with heredity, meiosis is pretty straightforward. The steps are pretty obvious. So I don't want to make it sound simple, but it's easier to study than heredity often is. So that's why we're going to go through more genetics today. Mitosis and meiosis are similar in the way chromosomes segregate, but differ in the number of cells produced and the genetic content of the daughter cells. So just have to remember that mitosis, you end up with 2 perfect copies of the original cell, and meiosis, you end up with 4 copies that are completely unique. In spermatogenesis, you end up with 4 different sperm, and in oogenesis, the making of eggs, you end up with just 1, and then 3 polar bodies. The separation of homologous chromosomes in meiosis 1 ensures that each gamete receives a haploid set of chromosomes that compromises both maternal and paternal chromosomes. That's crossing over. So when we see the crossing over happening and then the genes line up where they line up in separate ways, where like some of dad are on one side and some of mom are on the other. During meiosis 1, homologous chromatids exchange genetic material via a process called crossing over or recombination, which increases genetic diversity among the resultant gametes, unless, of course, they're too close together, in which case we're going to see what are called linked genes. Sexual reproduction in eukaryotes involve gamete formation, including crossing over, the random assortment of chromosomes during meiosis, and subsequent fertilization of gametes serves to increase variation. You will not need to know the different sex cycles of plants and animals. Thank goodness, because plant sex cycles are ridiculous. And I think I had to memorize them for AP Bio when I was a senior, and they were not fun. DNA and RNA are carriers of genetic information. It's important to remember that DNA is the one that carries most of our genetic material initially, but eventually we need to make a copy of it, mRNA, to make proteins. So thinking of DNA as the only genetic material is incorrect. Plus, there are things like viruses that carry RNA. Coronavirus, right now, what we're dealing with is an RNA virus. Ribosomes are found in all life forms. So all living things have ribosomes. All living things go through transcription and translation. It's just that the place and the process that are slightly different. And we'll talk about that in another video. Major features of the genetic code are shared by all modern living systems. It's one of our evidences of evolution is that we all use DNA. And by sharing that DNA, and we can even compare it, we can tell whether or not something is closely related or not. And the core metabolic pathways are conserved across all currently recognized domains. Basically, that's just saying like, hey, everything uses some sort of version of glycolysis, and a lot of organisms use like photosynthesis and things, and they share similar molecules. Now, before we get started, in the chat, does anybody have any questions about all the crazy review stuff? Because that's as fast as I'm going to go, and that's as much as I'm going to go through it now. If you do have more questions or you want to ask something one-on-one, by all means, email me, and I will make a video chat for you, okay? Fridays are great for that because Fridays are just prep days. Now, Mendel's law of segregation and independent assortment can be applied to genes that are on different chromosomes. This is incredibly important. Once they're on the same chromosome, if they're close together, there's a good chance they could also be linked, which means that they no longer follow these rules. Now, not everything's going to follow all these rules anyway, which is why the law of dominance is not included on my list. Because some genes aren't dominant, sometimes they get shared. Anyway, The law of segregation, only one of the two gene copies present in an organism is distributed to each gamete, egg or sperm cell that it makes, and the allocation of the gene copies is random. What this means is that if you have a parent, let's say we're going to use these letters, big R, little r, big D, little d, okay? And according to the law of segregation, only one of the two gene copies present in an organism is distributed to each gamete. So in the gamete, R can either go here or here. Okay, they can't be they can't be moved together. So they get split. So let's ignore the D. We don't need the D for now. So if we're making sperm, big R, little r, one big R goes to one sperm, the little r goes to another sperm. They are distributed evenly. They can't be gone together. Now, the law of independent assortment is the alleles of two or more different genes get assorted into gametes independently of one another. And basically what this says is if you have, let's say we're looking at a couple of different traits. When we see the law of independent assortment, it tells us that there's going to be 4 different combinations where big R can go to this one, and then it's going to get a big D. Big R can go with little d. And you can think of this like FOIL in math class. So first, outside, inside, last. And so these are the 4 different types of genes or gene combinations that can be sent onto the gametes. The reason for the law of independent assortment is because the chromosomes, when they line up in the formation of sperm and eggs, you have mom's chromosomes and dad's chromosomes, they line up opposite of each other, but they don't end up with like all mom and then all dad on one side or the other. Instead, what you get is you can have any combination of them. You can have a bit of mom and a bit of dad on both sides. And it's completely random. And each one that gets made is completely random. And so when we look at genetics problems, which are mostly about how we solve probability, what we're seeing is we're seeing randomness. And if they don't inherit randomly, things start to go a little crazy. And that's when we start to look for linked genes, which is something we're going to talk about later. So the big guy, Mendel. Mendel's traits follow the following rules. For every trait, there are only 2 alleles. That means that you can't have like like 3 genes controlling one thing. So human eye color is an example of that, where it's like 5 or 6 genes. For every trait, there will always be a dominant trait and a recessive trait. Generally, we use like big D and little d. Sometimes you'll also see a plus or a minus. And every once in a while, we see like really weird genetics problems with like fruit flies and stuff like that, where they'll use like VG plus and VG minus, okay? So just be aware that any one of these 3 things could be used. And every trait is controlled only by 1 gene. So you can't have more than 1 gene controlling like the color of something or anything along those lines. Mendel's P traits, by the way, it's kind of helpful to just kind of know offhand, like, hey, these are the traits that Mendel's P's follow. And everything doesn't follow this, by the way. Flower color can be different in other plants. So flower color, seed shape, pod color, flower position, and stem length are all examples of Mendelian traits in pea plants. And so what we see is all of these traits, he got really lucky, by the way, because if any one of these traits had been like sex-linked, or, which doesn't really work in plants, but like codominant or incompletely dominant, everything would have been messed up and he probably wouldn't have been able to come up with stuff. But just to be aware, these are Mendelian traits. They're going to follow just a normal Punnett square. The key vocab for the heredity unit that you might want to just be aware of and have a basic understanding of are things like dominant and recessive, where a dominant trait will show up over a recessive trait. A test cross is anytime you take a trait and you cross it with a recessive to see if the one that you're crossing it with, so like our test cross, if we want to know if something is heterozygous dominant or homozygous dominant, we would use a test cross to figure it out because the results would change depending on which one we get. Now that brings us to heterozygous, which is different, and homozygous, which is the same. Heterozygous would be like big D, little d, or homozygous would be big D, or little d, little d. The other words to be aware of with this are hybrid and purebred. Sometimes those words show up too. Genotype is the type of gene you have. So it is the letters that you have. I think somebody just tried to join and I didn't get a chance to add them. So I always like to think of it as just the letters or the genes that they have. The copies of the gene. The phenotype is the actual physical traits, what we see, what the protein is that's actually being made. Because you have to remember, when we're talking about genes, we're actually talking about a long code of DNA that's going to turn into a protein that's going to do some sort of job. It could be something like making a protein to control the color of your hair. It could also be a protein that controls like how ions get in and out of the cell. Each of these things is coded for by a protein, which is encoded by a section of DNA. So a phenotype is the physical traits. Other words to be aware of is like monohybrid, means just a cross between one trait cross. And then the dihybrid, which is a two trait cross. A P generation is the parent generation. Okay, and so that's going to be our initial cross. Our F1 generation is going to be our cross, what we get from our parent cross. And the F2 generation is a cross of the F1s. And a lot of times with genetics problems on the AP test, the way they're going to set this up is they're going to set it up so that when you get your F1 generation, all the offspring are probably going to be the same. A lot of times they'll be like heterozygous. And so that way, when you set it up, when they say, hey, and then this is our F2 generation, you can automatically figure out, oh, okay, this is from the F1. They all had heterozygous. It's a heterozygous cross. The last two words, and they're really important because they're going to be words that are clues if you run into a genetics problem on the AP test, is autosomal and sex-linked. Autosomal are going to be anything that's not sex-linked. And then sex-linked is going to be generally what's ever on the X and Y chromosome. And depending on what kind of genetics problem you're doing, the results end up being different. Now, I'm going to run through a quick example of a Mendelian genetics problem that could show up on an AP test. This is actually an AP question that has been modified to look a little differently. So a new species of fish was discovered off the coast of Brazil. Several different crosses were performed, each using 100 females and 100 males. The phenotypes of the parents and their resulting offspring were recorded. So this is the first cross. Cross one, true breeding male bluefish were crossed with true breeding female redfish. When we see true breeding, what that means is that they're homozygous. All the F1 offspring had blue scales, and the F1 fish were crossed, and the data for the resulting F2 fish are given below. So what we want to do is we can set up a basic Punnett square. I'm going to pick a letter. I'm going to use B for blue and little b for red. I'm not sure if this is going to be dominant or recessive yet. I have to play it out with the problem. But looking at the numbers, there's a lot more blue than red, which would suggest that probably blue is dominant. So I had a true breeding blue, which is big B, and a true breeding red, which is little b. And when I run my first cross, these are going to be my F1 fish. And they're all the same, big B, little b. And so by knowing that, these are my F1s, it says, oh, hey, here's the F2. My F2 Punnett square is simply just taking the offspring from the F1, which is big B, little b. Those are heterozygous. And I'm going to run them through my Punnett square. Down and across, down and across, down and across, down and across. And so what I end up with is I end up with 1 big B, little b. Oops, sorry, big B. Pay attention. All right, 2 big B, little b's. And because I'm not filling in my Punnett square very well, this should be 1 little b. From there, what I can do is I can figure out these two both code for blue because heterozygous is also blue. So I end up with 75% blue and 25% red. It says, what conclusions can be drawn from cross 1 and cross 2? Explain how the data support your conclusions for each cross. So in this case, my conclusions are, in cross 1, all offspring were heterozygous, which resulted in all the fish being blue, okay, which is where I got to this first cross. In cross 2, the heterozygous offspring were crossed, and so I should have ended up with approximately 75% blue and 25% red. And if I look at my numbers and I figure them out, I've got 375 plus 133 is 508. And if I take 375 divided by 508, I get 0.738, which is pretty close to 75%. And I'm pretty sure if I ran that with this one, it's probably close to 75% as well. And you might do this kind of calculation on the AP test and say, hey, it was 73.8%, which is close to 75%. In order to prove yourself correctly, though, you would have to do a chi-square test. And if the AP test were to ask that for you, they would give you the information you need to run it. In this case, we would be given this, and then we would be given the formula for it. Does anybody have any questions on the first page? If not, I'm going to move on. Now, everybody's favorite part of genetics is the probabilities and how you use them. There are a number of things that you need to remember for genetics problems. If you're going to solve anything with 2 or more traits, it's actually easier to use probabilities than it is to figure out a Punnett square. If you do a 3-trait Punnett square, you're looking at 16 by 16, no, 8 by 8. That's a lot of squares to fill in. 4 traits is 16 by 16, that's even more. Instead of doing that, you want to use probability instead. And the way to kind of look at that is if you can remember what probabilities you're going to get depending on the cross, or just do quick little 4 Punnett squares, you're actually going to find that these problems are a lot easier than trying to do an 4 by 4 Punnett square. And so what I mean by that is if I cross big D, big B, little b by big D, big B, little b, and I want to know what my genotypes and phenotypes are, instead of making a giant Punnett square and filling it in for 5 minutes and wasting all that time that I could be doing on other things, what I can do is I can use some basic probability. And by doing that, I'm just going to set up 2 quick Punnett squares. These are much quicker than the 4 by 4s. in order to figure out how percentages work, these are all working off of quarters and halves, which are really nice and pretty easy to work with, let's be honest. And so if I want to know, hey, what percent of these offspring are going to be heterozygous for both traits? And so I look at it and I go, okay, it's a 50% chance or a 1.5 chance of being big D little d, and a 50% chance of being, or half, of being big D little d. What I can do is I can figure out, hey, I can use what's called the multiplication rule. What is the probability of them being heterozygous for this and this? By using that, I can go, okay, to figure out how many are heterozygous, 1/2 times 1/2 is 1/4. Because you're just multiplying across. Okay, so if I actually did the giant Punnett square, I would find that it'd be 1/4. And so what you can do is you can figure these out. Now, if you want to know, hey, what's the probability of something being at least this or that, or you're looking for keywords, by simply knowing the percentages, it's a lot easier to work with these problems. And so another example would be like, as soon as you know if something is homozygous dominant and heterozygous, you end up with all the same phenotype, but a 50-50 chance of being big little and big big, okay? And by just remembering these percentages, solving mathematical problems makes it just a little bit easier for looking at Punnett squares. And you don't always use them. Like if you think you can get away with doing a 4x4 Punnett square and you want to, go for it. But understand that it takes a lot more time. There are a lot more problems like this on YouTube that you can watch to see how genetics problems are solved using probabilities instead of Punnett squares. I would recommend checking them out because they're really helpful in time saving. Now, if worse comes to worse, we'll go through some more of these on Wednesday because I know that the probability stuff is a little confusing. And yay math, right? So, all right. There are a couple of different kinds of examples of genetics problems that odds are aren't going to show up on the AP test, but I'd still like to kind of go through them real quick. One of those examples is what's called incomplete dominance. It's when the two traits, if you have both copies of the gene, one is like big and the other's little, instead of the dominant one showing up, the recessive one and the dominant one mix together, and you end up with a combination of the two, like mixed. And this is going to change how the probability works. So an example might be like if you have a red flower and a white flower, when they come together and you have heterozygous, they actually make the same, they make a different color. Now, it's up to you how you want to solve these. I teach the freshmen both ways and tell them to pick one. You can do R for red and W for white, in which case RW makes pink. So WW and RR. Or if you want to just use one letter, big R for red, little r for white. And then big R, little r makes pink. Okay. Now, if I have a red plant and I mix it with a pink plant, big R, I'm going to use R and W. It just makes things easier. And pink, RW. And I run my cross, I end up with %50 red and 50% pink. So when I do these problems, incomplete dominance ones tend to throw off the percentages. You're not going to get like 75-25. You're going to end up usually with 50-50 or 100%. The only exception being if you have two pink you end up with 25% red, 25% white, and then 50% pink. So understand that the percentages change a little bit with incomplete dominance. The odds of this kind of problem showing up on the AP test are pretty slim, particularly in FRQ questions. They're usually either like comparing an autosomal regular gene like dominant and recessive and sex linked, because those are the big ones. They're easy to test in a chi-square test. Codominance is another weird one, where instead of having a dominant and recessive trait, instead what you end up with is you end up with both traits showing up at the same time. So in a heterozygous individual, they come out as equal. An example would be like in a roan horse, where you have one color mixed with a separate different color, and you end up with like spots that are similar to both. This will also change your probabilities. Another example of codominance is blood typing. By the way, if you haven't heard, we are looking for people to sign up for the blood drive in Mayville. We need a couple more high schoolers. So if you haven't signed up and you're willing to donate, we'd greatly appreciate it. I mean, if you're worried about coronavirus, stay home. But like, we could use more donors. Just remember with blood typing, there are a couple of different kinds. But we're going to use I's as well as the blood type. So IA, IA, and IA, little I, are both type A. IB, IB, and IB little y are type B. IA, IB is type AB. And then little I is type O, where little i's are recessive. Okay. Odds are they're probably not going to ask you a blood typing question on the AP test in FRQ form. Okay. But it would be helpful to know them just in case, and kind of the generals of how they work. So if you have an AB person and they have kids with a type O person, what you end up with is 50% type A and 50% type B. Pleiotropy is where one gene has multiple phenotypic effects. And examples of this are cystic fibrosis and sickle cell anemia. The thing to remember with pleiotropy isn't so much that you need to know all of the things that it involves, but that sometimes a gene controls more than one trait in a person. In cystic fibrosis, it can cause a number of different effects, as well as sickle cell anemia is also like this. An example for sickle cell is that people with sickle cell anemia have a number of different issues with their health, including heart issues, as well as an increased risk of both stroke and heart attack. Because their blood is affected, but so is their heart, and from what we understand, it can also lead to issues in the brain. And then what's interesting about sickle cell, and we'll talk about it in a little bit, is if you're heterozygous for it, it actually creates a bit of a benefit for those people. But generally, if you're homozygous and you have sickle cell, it creates all sorts of health problems. Another example is epistasis. It's where one gene at one location can affect the gene of another location. An example is these cute Labrador retrievers. There are two traits that control lab color codes, or coat color. The first is E. E determines whether there's any color at all. If you're big E, you end up with a lab that has color, okay? So either black or brown, we call it chocolate lab. And then a little e is guaranteed to be a golden lab, okay? So these are yellow labs. So long as they have this little e, it controls whether or not the color shows up. If you do have a big E, it depends on what letter B's you have. So if you have at least one big B, you're going to be a black lab. And then if you have a dog that has two little B's, but still has at least one big E, you end up with a chocolate lab. And so that's where lab coat color comes from, which is pretty cool. This is an example of epistasis. So where one gene, in this case the E gene, controls whether or not the B gene shows up. They're probably not going to ask you a whole lot of questions about epistasis, but it's nice to review. Another example of a crazy non-Mendelian trait is polygenic inheritance. It's when two or more genes affect a single phenotype. An example of this would be like your eye color. Okay, eye color is controlled by multiple different genes, which is also one of the reasons why there are different colors of eyes other than blue and brown. Okay, there's like green and a whole bunch of other colors that I can't remember off the top of my head right now because I'm trying to get through all this. But understand, it's the same with hair color too. There's a bunch of different hair colors, natural hair colors, and so there's multiple different genes that affect it. Talking about human genetic problems are a couple of different things. So two recessive disorders are cystic fibrosis and sickle cell anemia. In cystic fibrosis, there are a number of different issues, but one of the big ones is it causes a malfunction in a protein that helps control how mucus is made in the lungs. And so what happens with people with cystic fibrosis is there's a bad pump in their cell membranes for making mucus. And so what happens is their mucus isn't made correctly and it ends up being extra thick and sticky. And so these people generally have a really hard time breathing. There are other things that it causes as well. But the big one, right, is understanding that it actually affects like how cells create ion channels and things like that, which is pretty interesting. For sickle cell, instead of a normal happy blood cell, you end up with cells that are kind of sickle shaped, which look like this. Now, normally our cells are happy. Think of them like inner tubes on a lazy river. They kind of bounce off of each other. With people with sickle cell, what happens is they end up with some of their cells or all of their cells in this weird sickle shape. And they tend to get caught on each other. So we end up with like a big old mess of sickle cells. And then it ends up forming a clot. So this is why sickle cell is oftentimes related to things like heart disease and stroke. And it's much more prevalent in the African American community for a number of reasons. And the main one is actually because it provides what's called a heterozygous advantage. In this case, heterozygotes, instead of making this or this, they end up making a mix. Because what it means to be heterozygous is you're actually using both genes. And so a lot of times with heterozygous, you make enough of the dominant that it just overlaps on the recessive, or the recessive just doesn't work. But you have enough dominant that it works just fine because it creates enough. In this case, they end up with both. Now, this still has some linked negative health effects, but what it does do is it provides a benefit against malaria. Malaria is a blood-borne parasite found mostly in Africa. And what happens is, for whatever reason, the sickle cell shape helps prevent malaria from infecting blood cells, which means that if you have a heterozygous version of sickle cell disease, you're less likely to die from malaria. And that's an evolutionary advantage for that area, so it's a trait that's stuck out. Otherwise, over time, there's a chance that it probably would have gone away. Generally, with negative traits, if they're bad enough, they tend to disappear over time because if you've got it, you don't reproduce and you die. Although a sickle cell usually kills you after you've reproduced, so that would keep it going. Excuse me, I'm going to take a quick break. All right. Huntington's disease is a nervous disorder. And it's a dominant disorder, which means that if you end up with just one of the two genes and it's dominant, you will end up with Huntington's disease. Now, the actual severity of it depends on the person, but generally what happens is it causes your nervous system to slowly but surely deteriorate. And the reason why this dominant trait has stuck around for so long is because it doesn't tend to kick in until your 40s or 50s. And so people with Huntington's generally have reproduced by then, and they pass that gene on as well. It's one of the reasons why it hasn't disappeared. Generally, if it's a dominant disorder and it affects you early on, and it prevents you from reproducing, over time, that trait will disappear. So those are the big human examples. These are the ones that like to show up on the test. A lot of times they're multiple choice questions. but I figured we should probably go through them just in case. For sex-linked traits, everything changes just a little bit. So instead of being on the other chromosomes, what we call the autosomal chromosomes, we are now on the sex-linked chromosomes. Now you have to remember a couple of things about the sex-linked chromosomes. If you have an X, it's a nice big chromosome, and the Y is a little chromosome. And because of that, there's only a few genes on the Y chromosome. One of these genes is called the SRY gene. It causes the human body to create male body parts. And so if there's something wrong with the SRY gene, you could be born XY and still end up developing female parts. If the SRY gene doesn't kick in, the X gene will just take over and start to develop female body parts. It's just kind of like on autopilot. The only thing is, without that other X to help with gene expression and things like that, what we see is underdeveloped female body parts. And because there's other genes on here, we tend to see some testosterone also being built. It's an example of a gender issue within biology. So you can be born as a male, but show up with female parts. And there was actually a runner from India who was in the Olympics, and she had some of her medals removed because she tested as male and she was in the female events. And there's a story behind that, and it's real. It's just, it's not common. Although we're not sure, it could be more common than we realize. There are two big types of sex-linked traits that you see in humans. The first is hemophilia, and the other is color blindness. Now, when we do a genetics problem for sex-linked traits, we're gonna have to remember we have to use our X's and Y's. So if you have like what we would call a carrier female, and I'm gonna use B for color blindness because C is a terrible letter for genetics. If she's a carrier, we would write her like this: X big B, X little b, and we'd call her carrier. Carriers carry the gene, but they don't have the actual phenotype. So she would carry the color blindness, but she doesn't have it. An example of this is my mom was a carrier. She can see color just fine, but she gave me an X little b, and I got the Y from dad because I'm a guy. And so what ends up happening is I end up being colorblind. So here's my dad, he's normal. Lucky him. Okay. Now, if I were to have children, here's mine, okay? Now, it depends on what the woman has. I could have X big B, X big B. If she has no history of color blindness, this in her family, this would be what it is. Okay, and you would end up with X big B, X little b for all the girls, and X big B Y for all the boys. So all the boys would be lucky. They don't have color blindness. They don't have to worry about it anymore. But the girls would all be carriers, guaranteed. Color blindness must come from mom if a boy has it. For a girl to have color blindness, X little b, mom has to be at least a carrier. She has to give up one X, and dad has to be colorblind. Okay, now mom could be colorblind too. Then you're gonna end up with all X little b's, so that would make sense. But understand that genetics problems are going to be a little different when you look at them than a regular problem, because now it's almost like doing a dihybrid because you have two traits to look at. You have male and female, and whatever trait you're looking at, okay? Now, hemophilia is a bleeding disorder, and it was somewhat common in European royalty. And what happened is there's a bad gene on the X chromosome because the X is much bigger, it carries lots of genes. And so what happened was Queen Victoria actually ended up with a random mutation that caused hemophilia. And she had a lot of kids. And the royalty, they tend to interbreed because to keep royalty within royalty, which is divine right, God gave it to them is what they believe. They married each other, and so a lot of cousins marrying and things like that. And so, yeah, it's pretty gross. And so what ended up happening was because a lot of her kids ended up with hemophilia, they spread it to their kids, or they died young. And basically what happens is your body doesn't form platelets properly. And so when you get cut, instead of your body healing and creating a clot and then eventually a scab, instead you just bleed, and then bleed some more, and then bleed, and then bleed some more, and you don't stop. And so what happens is if you have hemophilia and like you go to war, you're going to die a lot faster than somebody else. And a lot of these people, they don't live long if they live in a dangerous environment. Now, nowadays, we have like medicines to help with this, and you can go to the doctor's office or the hospital is probably where you're going to go if you get a cut and you have hemophilia. Color blindness is just a incorrectly developed sensor or gene, so a protein in your eyeball. So there are different receptor proteins in our eyes, and some of them see color and some of them see black and white. And so the color ones in a colorblind person aren't quite right. And so they end up not seeing colors quite right. And you've probably seen it in class because I've asked kids, I probably asked you guys, hey, what color is this? Because I don't know. Because blues and purples look similar and reds and greens look close. So color blindness, and this is pretty common actually. This is, there's quite a few people that have it. There's usually like 1 in 100 people. It's much more common in guys because it's much easier to get than getting 2 X alleles. Bar bodies in females, because guys, it requires 2 X's. In females, what happens is one of the 2 X chromosomes oftentimes will get shut down after a certain time. And so an example of this is like cats. In cats, bar bodies are what cause calico. Either you have black or orange. And so what happens is if the X with the orange is turned on, then you end up with orange fur. And if you have the X with the black fur shows up. And so you end up with a mix of both. For the probabilities in sex-linked traits, you just have to keep in mind, it depends on what they're asking. But for the most part, if you have like a carrier female and a normal male, you end up with this, this cross. Where you end up with 25% normal female, 25% normal female, but carrier. And sometimes you just mention, hey, they're a carrier. 25% normal male and 25% colorblind male. Okay, whereas up here, it was 50% carrier, 50% normal male. So this is what a lot of these end up looking like. And so their probabilities are a little different. And the reason I mention these probabilities is because eventually you're going to be asked to do a chi-squared. And so if you're looking at a sex-linked trait, and in this case, the males have it here, so 25% of the males have it, 25 don't, you could be like, oh, hey, 75% of the females were normal, but 25 were not. That tells you something else is going on. And so you just have to pay attention to percentages. So when you do your chi-square, it's like, oh, that's right. I'm doing a sex-linked problem. I need to use my X's and Y's and figure out my numbers from there. There are a number of practice problems, and I'm pretty sure we're going to be looking at one on Wednesday in the FRQ section. Now for some of the crazy, okay? Now, linked genes. Linked genes are located on the same chromosome and found close enough together to not independently assort. So early on in meiosis, in prophase 1, all of your chromosomes, they line up. So mom and dad's chromosomes, they line up. So here's, let's say this is dad, and this is mom, okay? The 2 chromosomes, they line up in prophase 1. And what's gonna happen is genes at certain points are going to cross over and they're gonna swap genes. So whereas before this was a copy of dad's gene and this is a copy of mom's gene, they are now completely new. They are new chromosomes. So this is a new chromosome. What happens with linked genes is during crossing over in the independent assortment due to Mendel, or not due to Mendel, but because Mendel explained independent assortment, these things, they shouldn't be connected. But sometimes what happens is genes are really, really close together. And so if a gene is right next to the B, it's much harder to swap it over separately. And so they get swapped together, and we call these linked genes. When we do a linked gene problem, what we're looking for is, does it come out in the same results that we got if we had them unlinked? So in an autosomal problem, and let's say you've got an autosomal problem where you'd expect, and I'm going to do two different ones because it's much easier to do this in a dihybrid. Let's go with a different letter than B and D. Let's go with B and E. If we did a hybrid of this, what we would expect is we would expect to have a 9-3-3-1 probability. And this is what I mean by memorizing probabilities. It makes it much easier. It's like the 3-to-1 when you have one example where there's just heterozygous. Like this is, 3-to-1. These are all dominant and this is recessive. But if we put them together, we end up with a 9-3-3-1, where this is dominant for both, recessive for one, dominant for the other, dominant for one, recessive for the other, and then all recessive. So we know approximately what we should get. When we run a linked gene problem, we're going to compare the percentages that we should get. So this is 9/16, 3/16, 3/16, and 1/16. And we're going to compare it to the percentages we get. And if those percentages match what's called the chi-square test, then we know that the actual genetics process is correct and they're not linked. If they are linked, it's going to fail the chi-square test. And then we're going to assume that the odds are the genes are linked. And then we can use some more math to figure out, hey, how are these genes linked? They're not going to ask you to do the linkage maps on the AP test this year, as far as I can tell, because that requires a lot more math and is usually more of like a multiple choice question. But that brings us to our chi-square test, which is everyone's favorite math question. I actually like these a lot more than I like Hardy-Weinberg, but that's me. So, so in a newly identified fruit fly mutant, Cyclops I, large and single in the middle of the head, is hypothesized to be autosomal dominant. Okay, autosomal means not sex-linked. The experimenter started with a homozygous wild-type female and homozygous Cyclops male. In this case, wild-type is going to be recessive because we think that the Cyclops I is dominant. And so the data from the F2 generation was 44 wild-type males, 60 wild-type females, and 110 Cyclops males and 150 Cyclops females. Does this data support or reject the hypothesis? Use the chi-square to provide to prove your position. So we don't actually need to do any sort of sex-linked stuff for this right now. All we need to do is figure out, hey, what is the probability for each? And so we're looking at autosomal dominant. This is our hypothesis. So the data from the F2, our F1 generation, by the way, let's go with E. So here's our F1 generation. They're all going to be big E, little e. Okay, so this is our F1. Our F2, our cross was big E, little e, because that's all we had, right? And so we should know that we should get 75% Cyclops and then 25% normal. And so what we're going to do is we're going to use chi-squared, which is observed minus expected divided by expected. So in this case, our wild type is going to be 44 and 60, so 104 wild type. Sorry if you can't read that. 104 wild type. And then 110 and 150 is 260 Cyclops. Our total number of flies, by the way, is 104 plus 260, 364 flies. We're going to need to know that because we need to do some percentages. So now we do our chi-squared. Our observed minus expected. Our observed is 104 wild type minus our expected, which is going to be 1/4, 25% of 364, which is 91. And then we're going to take this number and we're going to square it. We're going to divide it by 91. Okay. So 104 minus 91 squared divided by 91 is going to be 1.86. Now, rounding is going to be a little variable on the AP test. If they ask you to do a chi-squared problem, they're going to have an actual range of numbers that they're going to be looking for the answer. So if you're a little above or a little below, that's okay, as long as you're within that certain range. So that's my first one. That was wild type. Now let's do Cyclops. So observed was 260. 364 minus 91 is 273 is my expected. So 260, let's see that. 260 minus 273 is negative 13. Doesn't matter, it's going to get squared. I end up with 169 divided by 273, which is 0.62, I round it. And then what I'm going to do is this is going to give me my chi-squared number, which is 1.86 plus 0.62, which is 2.48. Now, to figure out my degrees of freedom, I need to take n minus 1, where n is the total number of groups. In this case, I've got 2. So n minus 1 is 2 minus 1. So my degree of freedom is 1. So here's the number I'm looking for. I'm going to go with the 0.05 because that's the one we always use in AP Bio class. 0.01 would be used more for statistical analysis in an actual lab, but for the most part we use this. Or I think we use a different number, a slightly bigger number. And we're looking for 3.84. Now my answer was 2.48. And so what that tells me is because this number is less than this number, I can accept the hypothesis I created before. In this case, I would accept what's called my null hypothesis, the original hypothesis. If for some reason this number was higher than this, what it tells me is that, hey, now something else is going on. If this is going on, I have to reject my hypothesis, and it tells me that there is something going on. In this case, I would assume that the two genes are linked. In this case, we're not looking at linked genes. Instead, I'd be saying, in this case, they would be sex-linked, okay? Because we're not looking at two different genes. We're looking at the X and Y chromosome and then the actual trait itself. So in this case, are we trying to figure out, is it autosomal or sex-linked? And we can say it's autosomal because our chi-square test, the number is less than this, we accept our hypothesis. Okay, I know it's a little confusing. Chi-squareds aren't too bad. You just have to remember that you're trying to get less than this number to accept your hypothesis. And if you get a number higher than this, it tells you something else is going on, okay? And yes, they do show up on the test sometimes. Linkage maps are going to take some numbers that we calculate. They're called recombination frequencies. And it's like a number line problem where they ask you to figure out where each gene is on the chromosome. Now, I've never seen an AP Bio test question like this, and my guess is they probably aren't going to show up on the AP test this year. And for the simple fact that it's a lot of math, and it's something that a lot of teachers don't always get to for linkage maps. But understand that basically they would give you like, hey, this one's from this length to this length, and this one's from this length to this length, and then you need to figure out what this one is in between. It's a number line problem. There are a couple of other things that I want to mention, and that's abnormal chromosome numbers and alterations to chromosome structure. Aneuploidy is anytime you have the number of chromosomes incorrect. In monosomic, you're missing a chromosome. An example of this might be XO, where a female is missing the X chromosome or a Y chromosome. In this case, she's still going to end up being female because she has an X, and the body automatically goes to female without the other letter. But generally, they're underdeveloped. And so we end up with a whole bunch of developmental issues. They're generally sterile. They have a bunch of other issues. There's generally some mental issues as well. Not mental, but like learning disability issues as well. Okay. Trisomic is when you have an extra chromosome. An example of this is trisomy 21, which is Down syndrome. Generally speaking, though, these two result in fetal death, a stillborn or a miscarriage. And the reason for that is the body is really complex and there's a lot of instructions going on. And without certain instructions, generally the developmental stuff stops happening, and the baby will miscarry. It's not the only cause of miscarriage, but it's one of the causes of miscarriage. Polyploidy is when you end up with multiple versions of chromosomes. And in animals, this rarely, if ever, happens. But in plants, it happens a lot. In fact, it's strongly linked to animal or plant evolution, where plants end up with more than one set of chromosome, and because of that, they end up it changes their species a little bit around. And eventually, because they're so different from their original species, they end up as two separate species, and eventually they just stop reproducing with each other. But for the most part, when you have abnormal chromosome numbers, with a few exceptions, you end up with the fetus not carrying to term. The last thing I wanted to mention is the alterations of chromosome structure. This isn't actually a mutation so much as it's actually a change in the whole chromosome. So mutation is like a change, like a small change in the DNA, where like a G has a substitution mutation to AAC. That's a mutation. These are alterations of chromosome structure. This is taking an entire chunk of chromosome and like deleting it, just getting rid of it, okay? Or a duplication where you take a chromosome and you make an extra copy of like the A part, and it's really hard to see. I'm gonna use a pen for this because it's hard to see. So it looks, so now it's too close. Okay, so here's my chromosome. So we're looking at a duplication. In a duplication, we have, let's say A, B, and C are our genes. In a duplication, what happens is, for some reason during like DNA synthesis, the A gets copied twice. And now we end up with two copies of A on the same gene. And this can cause a gene to go into like an overdrive where we end up with more protein than we need. Now, a lot of times these are really well controlled, but duplications can cause issues as well. In an inversion, you take the two letters and you actually take the letters and you're going to end up switching them. Now, this wouldn't be a huge deal, except for you have to remember that chromosomes are highly controlled during transcription and translation through gene regulation. And so there's generally a whole bunch of code before the protein that controls whether or not it gets expressed or not. And there can also be things like methylation, where the cell controls which genes are turned on and off by binding the actual DNA together. And so that's part of the structure of chromosomes. And that might be a good idea to review those. The last one is a translocation. In a translocation, what happens is an entire chromosome, like a chunk of it, gets swapped to a completely different numbered chromosome. Any number of these things can cause severe developmental issues within a cell and within a body. And a lot of times what we see with these is if you have any sort of major change to a chromosome, it causes major birth defects. And a lot of times it ends up also causing a miscarriage or a stillbirth. So these are all things that can actually cause a fetus to stop developing. They usually don't ask a whole lot of questions about this on the AP test, but it's nice to review it, not only because it's important and you never know what's going to show up on the test, but also because it's important to understand and know in the real world. So that's the quick and dirty of heredity. On Wednesday, I will be going through a whole bunch of FRQ practice problems with you so we can look at what's expected and how to read through and do them. I'm going to stop the recording now, and then for those of you who are watching live, if you so choose, you may ask any questions in the comments, or I can just unmute all of you.